I'm going to move toward the next speaker, Dr. Sovik Das. He's a assistant professor from IIT Delhi, and his talk is going to be about microbial electrolysis cell. Dr. Sovik, all yours.
Thank you. I hope my screen is visible.
Yes, I can see your screen. Yeah.
Okay. Yeah. So thank you for this invitation to deliver a talk for this session. So I am Dr. Sovik Das, and I work in Department of Civil Engineering of IIT Delhi. And today I'm going to talk about how electrochemistry and bioelectrochemistry can be helpful for the removal of contaminants, the emerging contaminants for the production of biohydrogen, and also for soil investment. So basically, we're going to talk about some sustainability aspects, some circularity aspects regarding the electrochemistry and bioelectrochemistry. So as we know, we generally now have changed our way of how we look into the resources or how we look into the waste. So waste is nothing but it's kind of a resource for someone else. So we generally try to make a resource from waste. So our group is basically called as a waste to wealth group. So we try to harness some wealth out of the waste, and in this way we try to align ourselves with the SDGs, the Sustainable Development Goals of UN, and we try to follow the SDG 6 and SDG 12 so that we can be cleaner India, or maybe we can do something for the environment and clean our waste in a proper way. So, as I told you, we basically work on bioelectrochemical and electrochemical systems. So, as we know, bioelectrochemical or BES basically are of various types. The oldest one I can say is basically microbial fuel cell (MFC), where it treats wastewater and it also produces bioenergy. Then we have microbial electrolysis cell (MEC) that our previous speakers were telling about that MEC. MEC is basically used for the production of hydrogen and also H2O2. So either you can produce hydrogen or you can produce H2O2 or you can produce methane as well. And it also treats your wastewater. Then you have one of the variations of microbial fuel cell known as the microbial carbon capture, MCC. So in the microbial carbon capture, we basically have algae in the cathodic chamber, and we generate the algal biofuel from the cathodic chamber, and it also treats your wastewater in the anodic chamber, and it also produces some amount of bioelectricity. Then we have the microbial desalination cell (MDC), as the name suggests, is basically is used to desalinate your brackish water. It is used to remove salts from your brackish water or saline water, and in the process, it also produces some amount of energy and some amount of wastewater treatment as well in the anodic chamber. And the most recent one is basically microbial electrosynthesis (MES). In MES, in traditional MES, basically CO2 sequestration takes place. CO2 is captured by the electrotropic microbes in the presence of some electrons, and they produce acetic acid mainly. Some biofuels are also produced, and it helps, and hence it helps in mitigating your global warming. So in and now coming to, I'll just switch on my this thing. Yes. Yeah. So now coming to electrochemical techniques, so we also use many electrochemical techniques to remove your waste emerging contaminants from wastewater and water, because unless and until you remove emerging contaminants from your water or wastewater, you are not able to use the water. You are not able to reuse the water for any purposeful conditions. So in electrochemical techniques, we have electrochemical oxidation, electrocoagulation, you have photo assistant methods, you have sooner electrolysis, electroreduction, electrolysis. All of these are basically used for resource recovery and your treatment of your wastewater. Then there are basically various reactions based on the various radioactive species that are there in the reactions, and they basically all, if you see, they basically end up in the pollutant. They basically target the pollutant, and they produce some intermediate, and ultimately we want to mineralize everything to CO2 and H2O. So that is what we try. We try to generate some ROS, reactive oxygen species, and they basically target the pollutant, and finally they break it down into some intermediates and ultimately do CO2 and H2O. We have seen a good number of publications on electrochemical and biochemical techniques for the contaminant remediation, like you see the dyes, you see the pharmaceuticals and the pesticides and other industrial compounds. They have been reported to degrade these contaminants with very high efficiency. So electrochemical techniques and biochemical techniques, based on what techniques we are using in which condition, they have shown good efficacy in removing these contaminants from your water and wastewater. So based on this knowledge, we try to use these techniques for further treatment of new age chemicals and new age contaminants that I will discuss. So if you see where this bioelectrophenton or basically bioelectrochemical techniques lie, so there are something called biological technique. We know like ASP, SBR, USB, they are basically the biological technique. Then we have something called the advanced oxidation techniques, basically AOP based techniques. And then we have something called the electrochemical techniques. And where these three system merge is basically where where we come up with something called bioelectrophenol. So it produces some electricity. It has very less production. It uses both anodic and cathodic treatment, and it has basically Fenton process at the near neutral pH. So, and there are challenges with the other techniques like the biological, advanced, or the electrochemical. But bioelectrophenol basically tries to look into the challenges, and it basically navigates the challenges and tries to give us a good quality of water, treating different kind of pollutants and reducing the amount of sludge being produced. So the main topic that was in the for the today's discussion was basically the biohydrogen production. We also work on biohydrogen production using MEC, as I was telling in the previous the slides. So what we do is that basically we generally try to use single chamber reactor. We have some double chamber reactors as well for the production of hydrogen using MEC. Yeah, as you can see here, we have a single chamber where we have a double chamber single reactor. We have a membrane in between, like this. If you see a membrane in between, and the protons move in and they produce hydrogen in the cathodic chamber. We are also integrating this MEC with the anaerobic system. It can be dark fermentation based system or any other anaerobic system from which the effluent from the AS system goes into your the anodic chamber, and here your the CO2, H plus and E minus are produced, and then E minus goes to the cathodic chamber, and H plus goes to the cathodic chamber to the proton exchange membrane, and you produce green hydrogen from this kind of waste. So we are trying to generate a good amount of hydrogen using different kind of organic waste. So this basically animation shows the new reactor that we designed and how it's getting operated. So you see the animation here. You have the anaerobic system, generally the dark fermentation based system or any other anaerobic system. The effluent from the anaerobic system AS basically goes into this chamber, and then we have the anode and the cathode in a circular way. And the reactions, as I told you, it occurs, and the hydrogen gas is basically produced, and it gets collected here in the headspace, and after that, it is basically converted, basically is conveyed from these pipes directly to the gas collection system, and it's basically a circular reactor that we generally use. So these are some small and big reactors that you're using in the MEC scale. These are small 200 ml reactors, and these are the big around 2.6, 2.5 liters of the reactor that you're using. We are basically trying to scale up this reactor from the small to big, and they have different configuration depending on different catalysts we are using, different types of the substrate we are using. We are trying out different combinations. We are also using nanotechnology for the degradation of plastics via biochemical and electrochemical techniques. So, as we know, India being the hub of irrigation and farming, we use a lot of pesticides and insecticides. So, currently, what we are doing is that we are trying to degrade these pesticides, these harmful pesticides, in the water and in the soil that is generally found in India. So what we are doing is that we are developing nanocatalyst from this different kind of invasive species. I will show you some of them, and from that invasive species, the nanoparticles that are formed, they are used as a catalyst for the degradation of these pesticides and insecticides in the bioelectrochemical and electrochemical system. So we are basically closing the loop of how the things are going on. If I show you, so this is basically a plant known as the Salvinia. So this salvinia plant is basically cultured in the wastewater. So we culture this or the grow the plant in the wastewater, in the real wastewater, and then we generate with the processing. As we know, we have leaf extract, then we have copper sulfate solution, and then we try to produce the copper nanoparticles. And these nanoparticles are basically uh, used as a catalyst for the removal of emerging contaminants. For this case, is basically pesticides from your water and wastewater. So basically, we are using wastewater as a resource for the growth of this plant. We are not using any nutrient culture and any nutrient media except the wastewater. And from this wastewater, we are trying to generate some nanoparticles, and these nanoparticles will act as a catalyst for the removal of toxic chemicals. So if I see, if I show you some of the results, we have got some of the results of the characterization of the nanoparticle. That we have seen. We have done the bacterial assay test as well. We have seen that this has good antibacterial property. It has some antibacterial property in terms of killing the microbes. If you see the halos being found here, and this is how we are trying to use this species for other applications as well. We did some characterizations regarding the same TEM, FTI, and all those, and we got good results for this nanoparticles. We are also trying to remove microplastic. We all know microplastics are a nuisance for us. They are basically some small or 5 to 0.1 millimeter size microbes or sorry, particles, which are generally of primary or secondary nature, depending upon from the source from which it is coming. And as we know, these microplastics has been found in blood, in human brain, placenta, and whatnot. So these microplastics are omnipresent. We can say they are present everywhere in the earth. In water, in food, they are present. So we are trying to find out the ways how we can remove the microplastic from this water and wastewater. For that, the quantification of microplastic is a major challenge. So hence we developed a protocol for the quantification of microplastic using electrochemical impedance spectroscopy. So we, as electrochemists, we basically use EIS to measure different catalytic property. But for this paper or for this work, what we did is that we use EIS for the quantification of microplastic. So as we know in EIS, we have solution resistance, your RCT, charge transfer resistance, and diffusion resistance. So in this case, what we did is that we try to quantify our solution resistance and quantify and link it with the presence of microplastic. And we are successful in doing that, not only for one plastic, we had done it for different types of microplastics, and it was giving a very good results. And you can see our paper that is already published where we quantified this method, and we use this EIS method for the quantification of microplastic. So this is a simple setup that we used for the quantification of microplastic. We have working electrode, reference electrode, and counter electrode, and then if you put your water containing microplastics and you run your EIS setup, it will give you basically the value of the RS, the solution resistance, and from that you can generate you already have a calibration curve, and from that calibration curve you can get the concentration of the microplastic. If we compare this, our method that we have developed with the other well-known methods, so our method is here the last, and if you see the cost, the instrument cost is very less, around 0.2, but if you see the accuracy, the accuracy of our method is around 95 to 96%, which is very high compared to the other method, which is in fact more costly or maybe similarly costly. So if you see the micro FTR or the micro Raman, all these things are very costly. You get around 2 to 2.5 millions dollars to spend for the instrument, but you get very high accuracy. But we can get almost similar accuracy, maybe even higher than that if you compare microraman with a very low cost, maybe 10 times less cost instrument. So that is what we are getting. And also we look at the analysis time. As we all know, the EIS takes majorly 5 to 10 minutes, not more than that. In fact, it takes less than 5 minutes. So if you consider your sample duration time as well, it is basically giving you highly accurate results in no cost and in no time. So that is what the advantage of using this quantification technique is for microplastics. We are also trying to remove emerging contaminant in terms of EE2. EE2 is basically, as we know, is basically a kind of estrogen. Is basically used for various medicines, and they are basically ending up in the water. And in the water, they are creating a problem for the fishes and other marine life. So we are trying to remove this EE2 from the water using electro oxidation. and we had some good results. If you see here, we optimized the condition. We got up to 80% of removal of E2 with very less energy consumption here. And this is also a work that we are currently doing. We are currently monitoring we are trying to increase higher the percentage of removal of the E2 using this electrochemical oxidation reaction. We also studied the toxicity analysis after treatment, and we did both EcoSAR and also the phytotoxicity in our lab, and we found that after treatment the toxicity is basically reducing. Hence, we can say that uh, treatment is active. It's not only removing the E2 from the water. It's basically degrading the contaminants and minimizing the contaminants from the water. We are also trying to remove PFAS. PFAS, as you all know, is basically a huge concern now. Now we have to try to remove PFAS from water and wastewater as well. We are targeting using the particle electrodes. So particle electrodes are basically a new kind of electrode that we generally use for the removal of emerging contaminants because they increase the efficiency of the electrochemical biochemical system. So we are trying to use particle electrode for the removal of PFAS because we all know that PFAS degradation is a major challenge, and PFAS in water is a major problem for all the countries starting from European Union to US. We are also focusing on parabens. Parabens, if you see, are basically very difficult to remove from the water. And the mineralization efficiency of other technologies into the physical, chemical, and biological are very less. So we are using electrochemical techniques for the removal of the mineralization of phthalates from water. If you see here, in just 15 minutes of time, we got around 100% of removal of methyl paraben and for different concentration from 5 to 10 milligrams. So as it is evident from the this work is that we are getting a very high removal, almost 100% removal in 15 minutes. And we also compared, so calculated the energy efficiency and the energy required for per meter cube of the removal, and it was very less. You can see the values are just in watt, is not even in kilowatt. So that is very less energy consumption for the removal of the parabens. We're also trying to remove phthalates. So phthalates are basically some contaminants which are there, which are used as before you produce plastics, or basically it's a precursor of making plastics. So hence it is basically removed. It's basically coming into the water, and we also trying to remove this phthalates from water. For phthalates, it requires very high treatment time. If you can see here, almost in 3 hours, for maybe 2 to 3 hours, we are getting almost 100% removal of phthalates for using a very less voltage of 6 to 8 volts. We are getting a promising result, but still we are trying to understand how we can improve that. We are trying to improve it by adding NaCl or by adding brine water with this, so that the conductivity of the effluent or the influent increases, and we are going to have higher production or higher removal of contaminants. So that is what we are trying to do. We are trying to increase the removal of different kind of phthalates. We are also trying to use or remove beta blockers. Beta blockers, as you know, is basically used for cardiovascular diseases. We are trying to use biochar. So the bagases, as you know, is basically a waste in India. We have a lot of bagases. We are doing this entire process, and we are starting from these bagases, and we are ending up in this nitrogen iron biochar, and this is basically functionalized in nature, and this is basically used as a catalyst. So we are again using some waste and generating some catalyst, and this catalyst goes into this reactor where we have different ways of removing your beta blockers. We are using your atenolol here in this system. So we have here multiple cathode anode system, and here we are trying to remove using this electrophenton and bioelectrophenton reaction, and this is basically a setup of an electrophenton reaction for removal of beta blockers. These are some of the setup that we are using currently for the removal of atenolol, and these are some of the tests we have done to study to characterize the production of your good catalyst, and using it for as a catalyst for the removal of beta blockers. We also try to see the intermediates form, the pathways, and how the toxicities are changing. And we found that these are the pathways through which, if you see, we are starting from here, and there are two main pathways, P1 and P2, and we are ending up here, and ultimately it is going into to become CO2, H2, and nitrate. So this is how basically we are trying to remove the different contaminants, mainly the beta blockers here. We also studied the toxicity and found that there's a good amount of reduction of toxicity for this test that we are doing. We are also using our electrochemistry or biochemistry to improve the engineering properties of soil. So as we know, when we have a lot of rainfall, we have a lot of landslide as well. We see a lot of contaminated ground as well because of pollution, but we also see a lot of landslides when you have a lot of rainfall. So what we are doing is that we are using electrochemistry to stabilize this ground and increase the strength of the ground so that the chances of landslides and this slope field is decreasing. So what we are doing is that we have sand or we have the soil. We are applying our electrochemical techniques. We are applying electrochemistry, electrokinetic improvement that we are doing. We are applying some anode and cathode, and you're dosing it with some electrolyte or wetting fluid, and then we supply some current, and then these depositions takes place. There's activity, electromigration, and electrification, and there is some deposition taking place, and taking place, and then we can see that electrification taking place, and as a result, we can increase the strength of this the soil, and electrolysis helps to bind the soil. So if you see our setup, this is the soil before treatment and this is the soil after treatment. So once the treatment has been done, the soil has become like a rock, and it doesn't move, and it's become completely steep, and it doesn't fall. And this is basically the sand before treatment. We apply electrochemistry and we get this kind of final product. Other selected research I just like to highlight in then this, and then I'll conclude. We are working with European Union for production of hydrogen using this dark fermentation plus MEC based system, where we are trying both the ways. Uh, for Indian partners, we are doing this Napier grass thing with MNRE. We are trying. So Napier grass is basically an energy crop, and that is basically mainly used for the production of methane. But we are trying it to use it in MEC, VAP plus in dark fermentation plus MEC for the production of hydrogen from this Napier grass. We are also trying to use food waste for the production of hydrogen using the same process. And we're also having algal based treatment for the purification of the gas that is coming, because it will have CO2 plus hydrogen, and there's some way of making algal biomass as well, and these algal biomass can be used again, reused back in the MEC as well as the cathode catalyst. We are also trying and working with Delhi and Indian government for the removal of different emerging contaminants in Yamuna. We all know Yamuna is very polluted, and it's always in the news for wrong reasons. First of all, we are trying to find out the PFAS and microplastics in the Yamuna, and then we are using our lab scale demonstration and then pilot scale demonstration for the design of a treatment plant, which can remove not only your inorganic matter, but also your microplastics and PFAS from your water. And last but not the least, I would like to thank my research group. We call ourselves Waste to Wealth Group, as you can see here, and this is our website. If you want, you can visit our website and to know more about our group. We were basically looking into, as I told you, on emerging contaminant resource recovery and some amount of catalyst production from waste. And I'm just presenting their work, what they have done, and this is their hard work that they have done. It's not my work; it's just their work that I'm presenting. Some of the collaborating institutes, like we work with many IITs and NITs in India, and there are some other universities that we work in India. And I would like to again thank other collaborating institutes like the international ones from Belgium, South Korea, Taiwan, UK, USA, and Thailand, to name a few. Then there are some government funding agencies like CPCP, DDA, and all those things who are working with us to help them and help in the removal of this emerging contaminant and resource recovery from different kind of waste. And we have an industrial partners. So again, thank you for funding our projects and helping us to work. So these are some of the references, and I think that's the end. Yes, and I'm open to questions. Thank you.
Thank you so much, Dr. Sovik, for a really interesting talk. Does anyone have any questions? We do not have time for a lot of them, but if you have like one or two, I can quickly ask them. Okay, so question for Dr. Velvizhi. What are the ANT reactors and what are the colored beads showcased?
Yes, please. What, excuse me?
Hi, Dr. Velvizhi. So the question is in the chat from Dr. Sharon. She's asking about what are the ANT reactors and what
Yeah,
are the color beaded
yeah, it is
reactors?
anaerobic reactor. It is like normal anaerobic reactors without electrodes. And those electrodes we use as a control for bioelectrochemical treatment with electrode and without. With electrode, it will be microbial PLCs or bioelectrochemical treatment. So ANT will be without electrode.
Okay, thank you so much. And another question from Dr. Sharon for Dr. Sovik. What type of interference may challenge the microplastic impedance measurement?
Yeah, so basically a good question. Currently, we are trying to use only water for quantification of the microplastics. But if you move on to some polluted water and wastewater, so the contaminants in the wastewater, the organic matter, they interfere with the microplastic detection because EIS, is very sensitive. So that is currently we are trying. We have tried only water, but we are trying with wastewater as well. That's our next step.
Okay, does anyone have any more questions? Can I quickly check something with you, Dr. Sovik? So you're using impedance spectroscopy. Are you trying to quantify the exact number of microplastic in wastewater?
We are not currently counting as such. Like many people, if you see they count the number of particles, we basically try to report it in terms of milligram per liter.
Okay. One question for both of you. The attendees want to know, would you guys be willing to share your slides within the group?
Yeah, fine with me. No issues.
Okay. Same with you, Dr.
Maybe I will check because some the works of patent I will remove it and I will share to you.
Okay, sure. Okay, so people, we will be getting the slide. Thank you so much to both of you for giving such an amazing talk. If anyone have any more question, please do reach out to both of them through social media or by dropping an email. Okay.
Thank you.
Dr. Sharon's have few announcement to make, so I'm just gonna give her the control now.
Yes, thank you very much, Dr. Sovik and Dr. Velvizhi, for your presentations. We thoroughly enjoyed them. And I'm going to give an announcement. So we are going to have additional seminars until the end of December. And Dr. Suvik and Dr. Velvizhi, you're more than welcome, and everyone who connected from India to attend them as well. They are public. And if you like to receive a calendar invite to the seminars, then please register yourself, your email, so that we can send you the calendar invite and it will facilitate your attendance here. So apart from that as well, I would like to announce a working table that we have arranged for Electrochem 2025, which will be held in September, the 2nd of September. It starts the 31st of August to the 2nd of September, and we have been accepted as a working table where we will discuss the present and future of environmental bioelectrochemical systems. In there, what we will aim to discuss are presentations from selected participants and discuss the state of the art of the bioelectrochemical systems, the future trends and environmental applications, and that will result on a position paper, a review paper that can be published, hopefully submit for publications. So I invite all of you to register to the conference so you can be part of this event. And right now they're still submitting, they're still accepting abstracts with the deadline today, but they will have special considerations for our working group. So there's still opportunities for all our presentations as well. So I'll invite you all to submit. And finally, the last announcement is our next webinar, which will be held on the 8th of September, and this is entitled Interfacial Bioelectrochemistry, Enzymatic Nitrogen Oxide and CO2 Reduction and Hydrogen Evolution, and is given by Dr. Cristina Cordas from the LabCat, the laboratory in Portugal, and we will share with you these posters for you to attend. So that's everything from my side. Thank you very much, everyone, for connecting. And I'm not sure, Maria. I'll let Maria finance proceedings. Thank you.
I would
Thank
just
you.
like to thank to the both of the speaker one more time. Thank you so much for presenting, and everyone, thank you so much for time for the attending the EBNet webinar. Please do show up on the coming one as well. Thank you so much, everyone. With that, it's a wrap.
Thank you. Thank you.
Thank